All right, Dr. Wohlgemuth is Vice President of Science and Innovation at Quest Diagnostics, and he's going to talk to us today about rapidly maturing technologies, which we've heard in many different iterations today. But his focus is going to look at diagnostics with high value for individual patients, maybe at risk of being lost in the specific scientific excitement that we've been hearing about. So losing the individual in this deluge of excitement and exuberance that we're experiencing.
Thank you. Do I need that? No. All right, thank you for having me today. Thank you, Larry, and the sponsors as well. And I like being at University of Colorado. I was reminded when my sister came here for college, my dad was a little upset with the tuition, the size of the tuition. She called 2 weeks later after starting and said, "This is great. I'm going to major in astronomy or astrology." And he said astronomy, and she said no, astrology. But I don't think we offer that as a degree here. But everything worked out for her. I have a really interesting perspective on this. I share all the exuberance. I actually do believe there's a lot of information in genomics and genetics and in proteins. And I've had a number of situations occur to me over time where there's a new diagnostic or a biomarker and it makes all the sense in the world. And you sit down with a physician and you say, here's what this test does and can do and has been shown to do. And the physician says, that makes all the sense in the world. And I really think that should be used clinically. But then it's not used clinically. Or 10 years later, it's still not used clinically. And so some of what I'm going to address is, when you have that in hand, why is it that a biomarker or test that makes all the clinical sense in the world is not adopted or used in the way that you think it should be? And I had that experience on an individual test where when I first got into diagnostics, working on heart transplantation and a blood test for transplant rejection. By doing that test, you reduce the number of invasive biopsies that cost $5,000 to $10,000 a piece. Makes all the sense in the world. So 10 years later, is it a part of the standard of care in heart transplantation? No. Is it being used? Yes. Then at Quest Diagnostics, we develop about 70 tests a year and put them on the market. And for each one of those tests, we have criteria of clinical value, clear clinical value for patients. Some of them are used and some of them are not used clinically. And so I'll try to cover, some thoughts around specific tests and also some of these barriers that keep them from being used clinically. So here's just some overview. The technology piece we've talked about quite a bit today, and I would just say it's necessary but not sufficient. And one of the major issues is that even when you have that test and the technology, it's the delivery of the information to the clinician at the time of the decision is a real challenge. And it's a technology piece which is both in the technology you're using to perform the test, but also in how you're delivering it back to patients and physicians. So I'll talk about that barrier. And of course, we need that information prior to accumulation of bad outcomes, tissue damage, and organ dysfunction. And then I think there's a major issue here where we get focused on an individual test, but then you begin to realize the physician or the patient doesn't care about one test. So if you're a patient with cancer, you care about all the tests in the continuum of care. You're also at risk for infection. And so you care about the tests that help you diagnose and monitor infection, and so on and so forth. So the concept of comprehensive offerings for patients and physicians is really important. So, this is just a simple, triangle trying to get to this value that we're all interested in for proactive or personalized medicine. We have to have a strong clinical need. There's biology and technology, and then the need for this translational research. And then there's technology again to deliver the information to the patient or physician at the right time. And we'll show you cases where some of these pieces have been in place, but you're missing one. If you're missing one, these things do not make it to physicians and patients and become a standard of care. So this is a lot of stuff here. I have so many of these fun examples. So I first want to get you excited and then maybe angry and frustrated and then maybe optimistic again at the end. So we've had a lot about what we're talking about here, proactive, predictive medicine and changing the paradigm for how we practice and take care of ourselves. And all these facts are really dramatic. To have a diagnosis of multiple sclerosis, you have to have 2 episodes of flare, which are documented in the brain on a scan, which represent irreversible neuron damage. So by the time you've had a diagnosis, you've had 2 flares which have damaged many neurons in your brain. Renal dysfunction, there's a 90% rule. Renal dysfunction and diabetes, you've lost the function of 90% of the important cells in that organ by the time we make a clinical diagnosis of the disease. Alzheimer's disease is a really interesting example. A lot of drug development in Alzheimer's disease right now. And by the time you have a clinical diagnosis of Alzheimer's disease, the accumulation of amyloid in your brain has largely occurred. So what is the likelihood we're going to develop a drug to pull out that amyloid and reverse the damage to neurons, which, by the way, do not regrow? So these are all sort of problem statements. And now I'll focus on where I came from. I'm a cardiologist, so we're the worst offenders, okay? So and when I went into medical school and was doing scientific work, I got real interested in doing scientific work in cardiology. But I ended up in a laboratory working on something down here, and it's really ludicrous when you think about it. Let's work on a gene therapy vector to help us with restenosis in artery after putting in a drug-eluting stent, which was put in at the very last stage of disease that has accumulated for 40 years. So all the research effort that I was involved with is way down here at the bottom, okay? But if you think about it, there are risk factors. There's a lifelong process of development of this disease. And you develop some atherosclerosis, you then have a heart attack, and then the invasive cardiologist comes in and puts in the stent. So the clinical care in cardiology and also the research activity in cardiology has forever been way down here at placing the stent and treating complications of stents. So I only do that so that you can't attack me for being a cardiologist, the worst offenders. This was in JAMA recently and is really interesting. In heart failure, we discharge patients from the hospital with heart failure. And there are all sorts of statistics. 1/3 to 40% come back within 90 days. And so in all realms of cardiology, we're just not very good at this proactive and preventative medicine piece. A lot of people have slides like these today. I've seen already that the burden of care or the burden of the huge healthcare spend that we have is on this late stage chronic disease and is not on the left side or the early preventative medicine piece. So why are we so reactive? I think first of all, it's hard to validate, to do translational medicine. It's coming, and I have some optimistic examples, but it requires not only the technology, but this translational medicine piece where the scientists and the clinicians get together to run validation studies that really make things a standard of care. We heard about financial incentives for physicians. It's very real. You're incentivized to do, if you put in a stent, you're incentivized by that financially, and you are not incentivized to do preventative care. There's no reimbursement mechanism for that. And then this one I'll talk about quite a bit as well, which is access to the information. So even if you have the greatest diagnostic in the world, if the information is not available to the physician at the time of decision making, it's a no-go clinically. The worst part that really bugs me is if it doesn't fit into the workflow. So you can get the diagnostic done, but it doesn't fit into clinical workflow, which is how the clinic is organized and how the patients are scheduled and But it's a real, it's a real issue, and that's what we hear when new diagnostics are delivered to patients and physicians. And then payers, it's changing, but some of the incentives for a long time have been on reducing short-term costs. And Steve Williams had a comment earlier on death not being, a valued or sustaining life not being valued. And I think this is changing on these incentives, but certainly there's been a focus on short-term as opposed to long-term. So now I'm going to show you just a kind of a variety of examples of tests which have been developed, which seem to be wonderful for one reason or another, but had some flaw or some barrier associated with them, which in some cases we've resolved and in some cases we have not. Fragile X, a very common cause of mental retardation, more in boys than in girls because it's on the X chromosome. And there's a fascinating molecular mechanism, which is expansion of 3 nucleotides repeats into long stretches on DNA, which causes this disorder. And we've been testing for it for a long time. And I'm going to skip forward to tell you that we screen now for cystic fibrosis. Prenatal screening is done for cystic fibrosis, but is not done for Fragile X, or not recommended by the committees that make these recommendations. And the reason, and if you look at the yield of screening for Fragile X, that yield is substantially higher than for cystic fibrosis. And in this case, the reason is ACOG, which is the main guidelines committee in this area, has said, yes, it makes sense. However, there are not technologies available that will be accessible widely to the population and enable testing of all the patients that need to be tested. And so going back then, we've recently, and others are now rolling out a new version of this assay, and it's very simple. The old assay required southern blots in every case to confirm the diagnosis. And for those of you in the laboratory, remember doing a southern blot. You get your gel and you run it out and you put it on a filter paper, and it causes a huge turnaround time in the clinical laboratory and a significant cost. And so by removing this with some technology innovation and using PCR techniques, we reduced the number of southern blots by 20-fold. The turnaround time is 4 days as opposed to 10 to 14 for most samples. We may have an ability to do this on blood spots and saliva. This was rolled out at the recent Genetics National Meeting, and the NIH has now said that Fragile X population-based screening is a priority for guidelines. So it gives you, a sense that it's not that simple. We've known about Fragile X forever, not forever, but we've had assays for it for a long time in the clinical laboratory. And on the surface, it appears that when you look at this yield of screening, we should have been doing screening for a long time if we believe in that. But there was a flaw in the ability to deliver the information broadly and in a quick manner to the population. Colorectal cancer, I think I'm showing this with a fair amount of pride that to address this issue of comprehensive testing, if you think about colorectal cancer, we have predisposition testing with genetic testing for hereditary syndromes, screening, diagnostic tests used on the tissue, treatment selection, which has been talked about today, and is a pretty good example of pharmacogenomics becoming real, and then monitoring tests. One important aspect is that we cannot get focused on a single mutation or a single test in this giant picture. In fact, there are 50 different test codes that we use, 50 different individual tests that comprise this continuum of testing for colorectal cancer. And the patient with the cancer and the physician caring for those patients cares about the continuum and not an individual test. So now I'm going to review every box on this diagram, gene by gene. But it helps you understand the complexity of it because this is HNPCC, hereditary colorectal cancer syndrome testing. This is one of 7 charts that we have that describe the flow of testing that occurs to work this up. a discovery is made in a laboratory for one of these MLH or methylation of one of these genes, and that's a good advancement, but that individual marker alone does not allow you to work up the patient. And in fact, you have to have the variety of tissue-based tests and then reflex down to the molecular test to get the diagnosis in the, all the patients with the hereditary syndrome. So another really interesting piece here. Colorectal cancer, we talked about this earlier, what do you do with information for early detection of lung cancer was one from Steve Williams. And we know in cancer in general, the way to cure a cancer is by early detection. In almost every case, if you can enable early detection, the cure rates go dramatically up. And this is colorectal cancer, and it's very true. Now, the interesting thing about colorectal cancer is we have screening methods. We have colonoscopy, and we have fecal occult methods, where you test a fecal sample for blood. And not surprisingly, the colorectal cancer screening compliance rates are less than 20%. Okay, so breast cancer, we have a nearly 60% compliance rate with screening. So interesting, we know if you can detect early, you can increase the chances of cure and reduce deaths very significantly. And we have modalities to do that, but the compliance rate is less than 20%. Now, some of you are eligible for colorectal cancer screening, and you're probably non-compliant. And so I think we can understand that there are issues with colonoscopy and fecal occult testing methods. A new marker has recently been validated, and I just want to give you a sense of this. This is the progression, if this is the lining of the large intestine from a normal lining to an adenoma, which is a benign growth, to an invasive cancer. And one of the early steps that's been known for some time to occur is altered DNA methylation in the tissue. And that's nice to know, but you can't do early diagnosis and detection of colorectal cancer on a tissue specimen. And so these methylation markers, these are adding a methyl group to certain stretches of DNA. These markers have been studied both in the tissue but also in the serum and in the stool. And so we've recently developed and released a test which is called ColoVantage. It's a methylated septin-9, and the important thing, it's plasma-based testing to overcome these compliance issues. There was a recent large clinical validation study of 7,900 subjects to show that essentially this test has similar performance to fecal occult methods, but is based on a plasma sample in the periphery. So the question is, how many lives would be saved if the compliance rate for colorectal cancer moved from 20% to 80% because it became a standard blood test as opposed to, I'm sorry to say this after lunch, 3 separate samplings of bowel movements with one of these tests? I don't have the answer to that, but it's a large number. And so again, it shows you a lot of things here. You know, even though colonoscopy and fecal occult methods theoretically can save a lot of lives, they don't because of compliance reasons. And then secondly, you know, this methylation issue has been around for a very, very long time. A clinical validation has just been completed, but I'm going to tell you, it'll be another 5 to 10 years before this is, you know, implemented in a widespread manner. Am I being depressing today? Is it? Okay. We talked about the mutation analysis of colorectal cancer. So I want to boil this down a little bit and get to this problem of physician education and how do physicians keep up with this information. So I've made it a little complex for you on purpose here, and that's because it is complex. There's a gene KRAS, and this is one we talk about. And when there are certain mutations in KRAS, a patient is very unlikely to respond to a very expensive therapy for colorectal cancer. And so if you know about this and you can test for those mutations, you can avoid that expense. That's nice, but also avoid spinning your wheels for 6 months and treating a patient while their cancer is progressing. Now, here's the problem. There are multiple mutations in KRAS. There are multiple mutations in these other genes, and this is just a partial list. Okay, so we had offered at Quest, for example, a test for each exon in each one of these genes. And we had the point earlier, of practicing oncologists, they haven't even heard of KRAS, right? So how is a practicing oncologist going to keep up with the very rapid pace of development of these mutations and understand which mutation do I test when and in what exon and at what time? Very difficult situation. So we actually took a set of 600 cases of colorectal cancer, looked by sequencing at all these genes and others for mutation rates, characterized the mutation rates in the cancers, and actually got that published as a letter in the New England Journal of Medicine, and then put it together in a pathway. And the way the pathway works is if you have a colorectal cancer and you're considering an EGFR antibody therapy, then order this test and we will test these mutations or the appropriate mutations in these genes. And if negative, there are no mutations, you're a potential responder. And if you have a mutation, you're less likely to respond, you might avoid. So this is a Band-Aid a little bit because it's the physicians at Quest and some of our advisors have helped us sort through the data and determine which genes matter in colorectal cancer. But then we have to do this continuously, going forward as these mutations change. And so we can do that, and we're sort of trying to make a clinical judgment on what's important here, but think about all the other mutations and all the other genes and who is going to interpret these for us and tell us what is clinically meaningful and not. The KRAS story is one of an obvious important marker that only, 10% of oncologists even know the name of. And then, however, it's much more complex than that because there's more genes and more mutations to keep up with. So by at least providing a simple mechanism for the physician to order these and get an interpretation, we might enable the use a little more. Okay, really interesting. So it was that Ed mentioned that there are mutations that occur in DNA, not just those that you get from your mother and father, but mutations that occur in a cancer as it's developing. And these are somatic mutations that affect the aggressiveness, the prognosis, and the treatment response for the cancer. But if those markers are in the cancer in a pancreas and you don't know about it, they're not helpful. Or if you know there's a cancer, but you have to biopsy it continuously to check on the somatic mutations, there's a severe limitation in that, of course, and that's not going to happen. So one of the areas I just wanted to highlight that may help make some of this information more useful and accessible is by measuring these mutations in circulation. So tumor cells die, they spill their contents out into the periphery, and these nucleic acids circulate. So we already know in leukemia that you can find the same mutations you find in the leukemic cells in plasma that's circulating. And so you don't have to go in for a bone marrow biopsy every time you want to know something about that leukemia. And it's actually interesting. In that case, usually physicians say, this isn't going to hurt. Or they're going to say, you're going to feel a little bit of pressure here. With a bone marrow biopsy, what they say is, this is going to hurt. Okay? So there's a severe limitation in our ability to use these many mutations we've discovered in the context of leukemia. But then, of course, in solid tumors, you cannot keep opening up the patient to get more tumor sample to access that information. So this has initially been developed in leukemia, and there's a lot of work now to make it available for solid tumors. And that's where the technology comes in again. For solid tumors, in order to be sensitive enough to pick these things up in the periphery, the next generation sequencing and some of the other technology platforms that are really coming along should make it doable. Okay, there's a continuum of care in gynecologic cancer, and I did want to cover OVA1. just to give you a sense of something that is I'd say a small success story in this picture, and also brings together this concept of technology and translational medicine. Maybe I'll even tell you a story of remaining resistance to adopt this technology. But OVA1 was actually cleared by the FDA, I think late last year, as a blood test to help determine the likelihood of malignancy in a woman who has a mass. So what happens is a woman goes in and has an exam or an ultrasound, and there's a cyst or a mass. We don't know whether that's a cancer or a benign tumor. The important thing is if that patient is sent to a generalist OB-GYN for the surgery, and it turns out to be a cancer, what will happen is the physician or the surgeon will start the surgery, realize it's a cancer, close the woman up, and send her for a second surgery to a specialist. So that's the double surgery and is a very bad deal. On the other hand, these specialist surgeons are in high demand and limited supply. And so you don't want to be sending benign cysts to have surgery by the specialist, which is a more expensive undertaking and a poor use of resources. So by developing this blood test, we have a tool to clean up that situation. And there's some facts here about how it better triages women to the right surgeon for their either ovarian mass or their ovarian cancer. And the specialist, the gyn-onc, who is the specialist, is specially trained to remove the malignancy. And in fact, it's been shown that outcomes and survival are better with the specialist if it's a tumor versus the generalist. However, the general gynecologist performs 2/3 of the ovarian cancer surgeries. So 2/3 of the time, the cancer is going to the generalist, where the outcomes are worse, and in many cases, they close up and send on for a second surgery. I'm not going to deal with the barriers on that one. It's too frustrating. I want to be optimistic. Okay, so cardiovascular disease, when you start to look at this type of framework, becomes a real mess, okay? And the reason it's a mess is because all of the disease states that we call cardiovascular are interrelated in some way. Meaning if you have hypertension, you're at risk for heart failure. If you have heart failure, you're at risk for arrhythmia. If you have diabetes, you get atherosclerosis, and then heart failure, and then arrhythmia. And so all these diseases are interrelated. And in fact, we believe in many patients there's a syndrome where these are in fact the same disease: diabetes, hypertension, and dyslipidemia, metabolic syndrome or syndrome X. And so in this case, it is critically important that if you're a physician caring for one of these patients, you care not only about the one test and the one twig of this tree out here, but the whole picture. And of course, if you're the patient with one of these conditions, you're at risk for or have some of the others. And so the concept of trying to put together comprehensive information for patients and physicians is important. Okay, so this one will be fascinating. Okay, there is Plavix, and it is maybe the #2 drug prescribed today, something like that. And I'm going to give you the short of it, and then I'll give you the long of it because it's a fascinating story. But this is a hit you over the head, gotta make sense indication. 40%, let's say, of patients who are getting Plavix are not responding to it well. So Plavix is used to thin your blood and prevent heart attacks and strokes. And we already know that 40% of the population has a mutation here, which makes this drug less effective. And we know that those patients with the mutations are at risk for higher rates of heart attack and stroke. And we know if you double the dose of Plavix or you switch to an alternative drug, you overcome this issue. Okay, 40% of patients are receiving a drug that's ineffective in them for preventing a very important outcome of stroke and heart attack. And so I'll just to give you the quickie anecdote, and then I'll go back through. I went to a leading cardiovascular center, and I said exactly what I just said there, and we went through all the data, and all the cardiologists in the room said, that makes all the sense in the world. I really, I think this is the best use of pharmacogenomics possible. This will really be great for patients. So we're walking out of the room, and the sales guy that was with me said, well, so are you going to start using it, or do you need information? I said, no, we're not going to. It doesn't fit into our workflow, really, and it'd just be hard to really do that because the nurses would have to follow up on the information. And so it's really a shocker. I got a little angry, and we debated over that, but it's remarkable. The physician knows how important this is and is educated on it, but then they can't do it, and why is not really that clear. But I'm going to show you a few things here to get to the bottom of it that I think, and the lesson here will be, even though it looks like you have it in your hands and it should help patients, there are so many barriers to overcome that you're sort of peeling the onion to the next issue that you're going to have to resolve. And so we've seen some of these before. This was the initial, or one of the initial genome-wide association studies. This is called a Manhattan plot, because this is the Manhattan skyline, and here's the skyscraper, which is the CYP2C19, which is a gene which was found to be associated with the response to Plavix. And it was a whopper. So this is the significance line, and it was obvious that this really makes a difference. And the reason is that clopidogrel, or Plavix, is a prodrug. And so when it's in the body, initially it's not active. It has to be converted to an active form, and it's converted to an active form by CYP2C19. So if you have a mutation in that, and you're a poor metabolizer, it means you're not converting Plavix to the active form, and it's not inhibiting platelets. On the other hand, there actually are some gain-of-function alleles where you can convert this too rapidly into active drug, and those are the bleeders, people who bleed when they go on the drug. So, I'm not going to go through this. There is clear data in 2 New England Journal articles and 1 in JAMA, which are our big journals, that say clearly if you're a carrier of one of these mutations, your outcomes are much worse when you're on Plavix than people who do not have the mutations, and much worse means more heart attacks, more stroke, more death. So the FDA has changed their labeling on this a number of times, and most recently in March put a boxed warning on the Plavix label that said, hey, clinician, wake up. There's genetics out there for this, and 40%, although we debate the numbers, a large proportion of subjects receiving Plavix are not responding. And there are genetic tests available, so maybe you should take notice. I can tell you now, so I'm not totally frustrated today, that in fact this is starting to be used much more commonly in a clinical sense. But I'm going to also show you some of the barriers. I'm not going to do the data here. I already mentioned that if you have a patient with a mutation and you double the dose of the drug, you overcome the issue in many cases. Or you can switch to prasugrel, which is a recently approved alternative drug that is not impacted by CYP2C19. And then there's the bleeders that I mentioned, that if they have a certain mutation, they're at increased risk for bleeding when put onto Plavix, which is also a very important piece of information to have. And so we've developed a test, which is a laboratory-developed test. It has certain of the mutations, and we're adding this star 17 for the bleeders. And we validated on saliva, and I'll tell you why that's important. So let me get to a couple of the barriers. We run into a lot of situations like the one I talked about at that leading cardiovascular institute, where they said, this is great, but we're not going to use it. And one of the issues is this. This is the time frame for the clinic visit. If you have a patient on day 0 who is going to have a stent put in a coronary artery to open it up to provide blood flow to their heart, they go on to Plavix at that point to prevent a heart attack from occurring after the procedure. And actually, they sometimes stay on Plavix for life, or at least for a year in most cases. So the problem is, if you have a blood test that's done in a reference laboratory and the blood is drawn on day 0 and the result is back 4 days later, then the patient was not put on the right dose of Plavix or the right therapy for this 4-day period which happens to be the highest risk period for a heart attack after a procedure. So that's not optimal. With a saliva method or just with clinic logistics, we are seeing clinics work a little more proactively where a saliva sample is sent in 4 days in advance of the procedure, or a patient goes to a service center, gets the blood drawn 4 days in advance of an elective procedure. The result is in hand at the time of the stent placement, and the right drug can be used. Now, of course, this doesn't work when it's an emergent procedure. Someone comes into the emergency room with a heart attack, you're going to be in this situation. And so this is one of the barriers. So let me talk about time and how we're addressing that. So far I've gotten as far as saying this is a winning application. We have issues with clinical logistics. It is starting to be used, but there's this remaining barrier of the information is really not in the hands of the physician when they want it in every case. So now I'm going to talk about time, which was our theme. This is 2009, some events that occurred here. The only reason I show you this is as a segue into here. These are all critical events, of course. The crash of Tiger's car into something in a Florida neighborhood. I just have to say, does this seem like a really long time ago, or does this seem like yesterday that this landing, that was the beginning of 2009 here. We had an election of the new president. There's Sully. We had the H1n1 flu. That's my hook, the only reason I do this. I like to look at this thing and say, God, time is moving fast, isn't it? Why isn't that test being adopted? It's been 1.5 years. In clinical terms, that's nothing. I'm going to come back to a timeline for you. You can guess what's in Q1 2010, okay? So if you look back last year, we had the swine flu, H1n1. And I call it CNN Day, because that day, all day long, there's a continuous CNN story about swine flu in Mexico. And we developed a laboratory-developed test. From April 26 to May 11th, we produced a laboratory-developed test for H1n1. Yeah, we're very happy about that. However, how useful is it when a patient comes in with flu symptoms to the hospital and you have to decide on therapy and whether to notify or label them as a flu patient to know 3 or 4 days later their status? And so what I'm not showing, we then went on to get FDA approval for this, and then a few months later, we put it onto a near-patient platform. Okay, so now we responded to H1n1, but the real need is for something in the hospital where you can get the information quickly. We put the testing components onto this disc. We put this coffee maker size thing into the hospital. And then we allow the hospitals to do testing for same day or maybe like a 4-hour turnaround time. The actual assay takes about 30 minutes, but with all the logistics, 4 hours. So that's important. We have to take tests like CYP2C19 for Plavix and many of the others that we've talked about and get them to the patient at the time of the encounter, or to the physician at the time of clinical decision making. So the H1n1 was the training ground. The Plavix assays will be on formats like this, which will allow those cases where you need the information immediately to deliver that information to the patient. I'm going to cover just a couple other things here, just to give you a sense of where we're going. In infectious diseases, we have microorganisms, and those organisms have genomes, and we can sequence those genomes and then do a lot of things with that information. And we're doing a lot of that now. However, the barriers there are cost and turnaround time. So the cost of this sequencing has to come down, and it is. And then the turnaround time has to be reasonable. If you're in a hospital with an infection, you're not sending out for 4 days waiting for a result. And so in this case, although we already know that the information in the organism's genome is very valuable, in order for it to really be used clinically, there's a technology barrier that is being overcome that would enable this to be useful. Okay, this is a little unwieldy, but it used to be, we talk about this model about the next 20 or 30 years we'll be moving that direction, but it seems like it's moving more quickly. And that is the old model for genetic testing, let's say, is that you identify that there's a patient that has some risk or some reason to be tested. You perform a molecular assay, and then you interpret and report that assay. And we do a lot of that type of testing in reference laboratories. We now believe that the future will look like this. For those diseases where the genome is important, an individual would be sequenced early in life. That information would be maintained in that patient's personal health record. We would need a reference database that would tell us what does that really mean, so that when the patient is being prescribed drug X, you refer to the database, you realize they're a poor metabolizer, and you actually have 0-hour turnaround time, because at the time of the event, all you have to do is compare the genome to the reference database and then provide the interpretation out of the database. So who has the database? Who is the judge of what is clinically meaningful or not in these mutations? Many, many issues to get there. But you can see, as far as making use of genetic information in pharmacogenomics, we believe there are tests now that are useful in this world. But if we can get to a point where we actually maintain our genetic information and compare it to a reference database, then it's not about performing the assay quickly, but about just having this information on hand at the point of the patient interaction. Okay, can someone guess what's in Q1 2010? Anything big happened so far this year? What's that? The Gold Symposium. No, not quite. It was. Now, I have to say, I did put a censor bar on here only. I just wasn't sure Larry would be okay with the uncensored version. We had of course, an important Super Bowl victory, and then Corey Haim passed away. It's a big news in Los Angeles, so probably not as big here. Okay. Time is flying fast, right? We have these tests, and we're discovering them constantly, and they look incredibly valuable on paper. The clinical indication makes all the sense in the world, right? And then we realize there's the next barrier, which is delivering the information to the patient at the time of the encounter, or that the information is not useful unless it's in the context of other information the physician or patient desires to have. Or many of the other barriers that I talked about today. And so no single test delivers the full value, and we've talked a lot about that today. Some of that's multiplexing, some of that's about information technology, bringing all the information together. And technology innovation that we've talked about allows us to detect key biological events, and technology innovation will allow us to deliver the information at the right time. So technology is not only in the assays, it's in the delivery of the information. And you know, the sequence-based diagnostics are moving to a point where diagnostic industry needs to become a IT industry or a provider of information and not a provider of wet lab services. So where the sequencing occurs, I'm not sure will matter in the future, but how it's interpreted and delivering the information really will. And so I've talked about the requirements for enabling proactive medicine. I've pretended to be very frustrated, but I'm actually very optimistic. Many of these things are actually making their way into clinical care, and I think our challenge is just to keep in mind when we think we're there, that we're probably not there, and to try to address the many barriers that are between the clinically relevant application in the marker and the actual use in the clinic, which has been much of our discussion today. So thanks for having me, and I'll take any questions.
I think you need this.
There's one here.
Thanks. Can I? Hi, Jay.
Hi.
Hey, so 2C19 and Plavix, and I believe it's Lilly is the maker of Plavix?
I think that's right.
So what's been their involvement in
Yeah.
in promoting companion diagnostics and the pharmaceutical benefit managers and the government in terms of
Okay, interesting.
happen?
I think in that particular, I don't want to generalize because in some cases, pharmacogenomics are actually really important to the pharmaceutical industry and they know it. And it's an important part of drug development or offering the drug to patients. In this particular case, I think the debate is around which of the genes are relevant, and I quote 40% rate of carrier status, which is true, and the pharmaceutical industry would say 5%. And so that'll tell you where they stand on this, is they're not enthused in this particular case about these mutations. However, the reality is that 40% are carriers who do not metabolize the drug to the active form. So I'd just say you got to take it case by case. In this case, I'd say they're not enthused because it's not enabling of their drug development and use. In other cases, they know that they have to use these tools in development or to actually identify patients for drugs. So it's a mixed bag on a high level.
It seems that you might have not explicitly mentioned one other barrier to getting these tests to the physician, and that is the large number of tests that you and presumably other diagnostics companies are actually building. It reminds me of the software manufacturers that come out with a new version of their software every 15 minutes and they expect you to buy each one of them.
Right.
I'm wondering if you couldn't actually succeed more by having fewer tests.
Yes. Yeah, there's a couple things there. First of all, I'll sometimes meet with a clinician and we'll talk about collaboration, and then I'll realize we already have the test on our menu that we're talking about, but nobody knows it. So the first part is when you have 4,000 tests on your menu is an awareness issue. The second part is they're often not organized in a way that's clinically meaningful. So I talked about the KRAS. Originally, that was organized in a way of mutation X and exon Y of KRAS, which is really unhelpful for a physician. They can't make use of it. So that effort was just simply to organize it in a way that is around a disease and not around a mutation, that provides a clinical interpretation for colorectal cancer management. And so some of it is about just making sure it's clear what it's useful for clinically. And then in other cases, perhaps you're right. I think we try to be cognizant of what is clinically validated and what is clinically useful and not. And we do not put tests on the menu that we don't feel are clinically useful. But there are many ordering physicians and stakeholders and patients out there who may want access to certain tests. I think the main thing is organizing them in a clinically meaningful way and never putting something on the menu that doesn't have clinical value in our view.
Thanks for a very nice talk. I'm Stephen Ack. I should identify myself as being with Eli Lilly and Company. We make Prasugrel, not
okay,
Plavix.
good. Clarification.
We make the better one. Oh, yeah. So I think you raise an interesting point. You talk about the cardiologist who walks out of the room and is not clear he's going to order the test. I think that same issue occurs in the whole value chain of drug development. From my own personal perspective, there was a lot of discussion at Lilly and with the development co-sponsored Daiichi Sankyo as to whether or not the genetics would even be studied. As you recall, when the two back-to-back New England Journal papers came out, one was a Lilly-sponsored investigators, and was a completely separate group, found essentially the same results. But the prevailing wisdom at the time was, don't ask, don't tell. Just develop Prasugrel. Why worry about the genetics of clopidogrel? We'll just have a better drug and have all of the $8 billion market that used to be theirs. I made that up, but it's a good story. But I'm sure some people are thinking that. I think the real issue is going to come when we have to choose between inexpensive generics, and Plavix will be generic soon. There already is in some countries. and the more expensive product, which we sell, which isn't going to be generic or cheap soon. And there I think the healthcare economics comes to the forefront, because you want to be using the best resource for the right person, but not the most expensive resource for people who don't need it. And I think that kind of decision-making adds a level of complexity that even goes beyond what your cardiologist is not dealing with already.
Right. A couple really interesting points I'll respond to is that the Plavix will be generic next year, and the price will come way down. And so the doubling of the dose of it won't be as big a deal, and the desire to stick with it might be higher as opposed to switching to a more expensive alternative. So the genetic information would be very important for identifying those that truly need the step up in therapy or the more expensive version of therapy. And you know, in the drug development side, I would say I think we're moving toward a time where it will not be acceptable and may not be acceptable from the FDA to run drug development trials without collecting the samples to do the pharmacogenomic analysis. And in fact, there's been some guidance put out there by the FDA already to say DNA should be collected in all pivotal trials going forward, because it's a huge shame when a pivotal trial occurs and we don't have the materials to even ask the question whether there are responders or non-responders based on biomarkers. And those trials are the only way to ask that question, because it's the only source of a powered, prospective trial. So I think more and more companies such as Lilly and others, they're going to do it because they want to do it and they know they need that information. But I think then the public and the FDA is not going to allow companies to not do it at some point in the future.
One more question.
Yeah, I have a question about adoption. And you had talked about how physicians had agreed with you that this is an exciting diagnostic test that has a role, et cetera, but they're not adopting. And then you alluded to the notion that it just simply didn't fit into, for lack of a better term, the workflow.
Yeah.
Now, are you really saying that there isn't the financial incentive? Or are you saying that there's too much inertia in the institutions and everybody is so protocol-driven that any variation, they can't even contemplate variation from the existing protocols?
Right.
What are you saying there?
A couple things. First of all, this testing is now being used, it's really taking off, not just for Quest, but in general, after the FDA black box warning and some other events recently, the Plavix testing is happening. I just use that anecdote as an example of, how could that be? I think in that case, it may not. There are many other diagnostics where there are perverse financial incentives that do not incentivize the physician to use the diagnostic. In this case, I think it was truly what I call workflow, clinic workflow issues of how do I do this, and then I'm going to have to call each patient afterwards. And actually, there is no incentive to do that, so there isn't a financial incentive to call them back. And it's just a whole new burden on the clinic and the staff, and they're overworked and very busy, and they just don't want to go there. So I think in that particular case, it's truly how am I going to tackle this and change my clinic to accommodate these new tests.
Thank you very much. Great talk.